Okay, so we are going to start with membrane transport. And let me get my screen shared. Me iPad. Me iPad is coming on. God, I'm such a dork. Okay. All right. So last time we left off on osmosis. Remember those hypo, iso, and hypertonic solutions. And now we're really talking about membrane transport. So when I mean membrane transport, I'm really talking about transportation through that phospholipid bilayer. That, remember, each phospholipid is amphipathic when we have polar heads and non-polar tails. So membrane transport can come in two ways. You can be passive or it can be active, all right? So passive diffusion is where something can diffuse from a concentration of from a high concentration to a low concentration, all right? Right through the plasma membrane. So diffusion, remember, is just going from high concentration to low concentration. Passive transport is diffusing across the plasma membrane. And so the two or three things that we talked about that can directly, passively just diffuse through are gases, O2, CO2, we're going to talk about nitric oxide sometimes, and those steroid lipid molecules. So they can go straight down through the plasma membrane, but remember, they are always going from a high concentration to a low concentration. If, for example, there was a lot more of these gases, for example, in the cell, they would then diffuse down their concentration gradient out. And I always think of transport, passive transport, is you're starting on the top of a hill, you roll down a ball, woohoo, no problem, easy breezy, takes no energy, right? So passive transport takes no energy, right? It just happens using the energy that's kind of stored in that concentration gradient. Oh my. Decline that phone call. Okay, active transport, on the other hand, requires energy. So this needs some sort of input of energy. Oh, sorry, my face is blocking. Needs some sort of energy, right? And that is because here you are moving against the concentration gradient. So instead of going down the concentration gradient, you're going against it. So you're going from low concentration to high concentration. And that's hard. That's not the way that diffusion normally works. So you have to have an input of energy. And I always think of this as kind of pushing a ball up the hill. Here you have high concentration, here you have low, and we're gonna force things in that direction, even though the things want to diffuse the other way. So you need a lot of energy here, all right? And if you're thinking energy in the cell, you should be thinking of that one special nucleotide that we mentioned before that is the currency for all energy in the cell, and that is ATP, right? So looking at passive versus active transport, again, passive, you're going down the concentration gradient through a plasma membrane, no energy required, right? And there's two kind of ways that you can have passive diffusion. One, you can diffuse directly through the membrane, as shown here, right? And then two, you can still go down your concentration gradient, but you can go through a protein, all right? And that is shown here, number two, and that's called facilitated diffusion. For active transport, we are going to see that, again, that is against the concentration gradient. You need to use energy, and we're going to look at two different ways you can do that, all right? Either kind of through proteins or through bulk transport, which we'll describe in a bit. Ooh, nice, I have two of those. Weird. All right, so I guess this is for passive. So passive diffusion we already talked about. So the only molecules, the types of molecules that can flow down their concentration gradient through a membrane are the gases, O2, CO2, NO, and the steroid lipids, okay? Facilitated diffusion, on the other hand, still going down their concentration gradient, requiring no energy. However, these molecules, for example, would be either polar or charged or large. So they can't go directly through the plasma membrane, right? They get stopped by that non-polar tail barrier. So instead, what they're going to do is they're going to just use a protein, a membrane protein. So this is a membrane protein. You're going to use that membrane protein just as a vehicle to get across, all right? So some of the protein, so you can use a membrane protein. And you can use that membrane protein, you can have two different kinds of proteins for facilitated diffusion, right? You can have what's called a protein channel. And this is really a protein that just kind of has a hole through it. So if you kind of look through, imagine something like this, where molecules can just go through. They just have like a pore through them. And aquaporins are an example of that. The other kind of proteins you can have are carrier proteins. And carrier proteins change the shape as molecules go through. So it's not just a pore. There's actually a conformational change, but again, that's not taking energy. So again, you're going from high concentration to low concentration, but you're using a membrane protein, and that membrane protein can change its shape as a protein or a molecule comes through, right? So again, we have gone, so for passive diffusion, no energy, down its concentration gradient. You can have diffusion right through the plasma membrane, or you can use facilitated diffusion, either using a channel or a carrier membrane protein. All right, now we're going to focus on active transport. Again, when you think of active transport, you should think of ATP. You should also think of going against the concentration gradient. So again, when you think of this, you should think of our good old friend nucleotide, ATP, right? So ATP, again, remember nucleotides have a 5-carbon sugar. They have a nitrogenous base, shown here. And in this case, it's adenine, that's why it's the A, and it has a phosphate group. However, ATP has 3 phosphate groups, and that's why it's called ATP, because this T means triphosphate, adenine triphosphate. When you have, you can add water, which is, what is that reaction? Again, it's called hydrolysis, and you can break off one of those phosphates, right? So now you have one phosphate that has been broken off, and now you have ADP, meaning adenine diphosphate. And with that, you get a release of energy, right? So energy is released when these kind of high energy bonds of the phosphates are broken in a hydrolysis reaction. So again, ATP plus water gives you ADP plus an inorganic phosphate plus the energy that we need to go against a concentration gradient. So active transport goes against the concentration gradient, and it requires lots of energy. An example of this that we're going to come back to several times is called the sodium-potassium pump, right? So the sodium-potassium pump is a really cool active transport because it does 2 things, right? The first is that it uses ATP to move 3 sodium ions out, all right, against their concentration gradient. So there's a lot of sodium already out here, and we're pushing more out. And at the same time, this cool protein, membrane protein, also moves 2 potassium inside, also against their concentration gradient. So there's usually a lot of potassium here, and we're pumping more in. So this pump uses ATP, and you can see 3 sodium out, 2 potassium in, both, here's the membrane, both going against their concentration gradients, right? And that is going to take ATP to do that, right? Another example of active transport is the proton pump, moving hydrogens against their concentration gradient. And we are going to see this in both mitochondria and chloroplasts coming up. So here we have in this diagram here that purple membrane protein is a pump. It takes ATP, the energy from ATP hydrolysis, to drive protons against their concentration gradients. Sorry. So you can see there's lots of protons out here already, and we are going to pump more there, more into there. All right. And what we're going to see is that in the mitochondria, we're going to see a gradient where hydrogens get pumped into this dark gray area, right? Called the intermembrane space. And in the chloroplasts, what we're going to see is that we're going to pump hydrogens to create a gradient in those thylakoid membranes, those kind of stacks of membranes, all right? So in these, when we're using these proton pumps, what we're doing is creating an electrochemical gradient, all right? Why is it electric? Because we have a charge difference. We're adding pluses. Why is it chemical? Because we are actually adding more hydrogen atoms, right? Another kind of active transport, and this one really confuses students, so we can go over this as many times as you guys would like in class. But here, just look at it one at a time. So look first, I'm drawing kind of a little dashed line, or actually I'm just gonna draw a big old solid line. First, we have a proton pump, all right? That purple proton pump is going to use ATP, and it's gonna pump hydrogens across against its concentration gradient. So we get a buildup of H's over here, right? That creates a concentration gradient, right? We know that things like to flow down their concentration gradients. So what we've done is we have added what is called a co-transporter, where hydrogens are flowing down their concentration gradients, but at the same time, they take a partner. It's like, giddy up, guys. Let me grab a sucrose. So at the same time, they, this transporter grabs a friend and brings it along for the ride. And that, in this case, in this example, it's sucrose, right? So what we've done is we had inputted energy at the start to create that hydrogen gradient. Then we're using hydrogen gradients, which like to flow down, to grab sucrose across, right? So plants commonly use the gradient of hydrogens generated by proton pumps in order to drive the active transport of nutrients into the cell, right? So this is kind of, this whole thing is kind of one system in order to get sucrose across, right? It's kind of a crazy way. We're using ATP to create a gradient. That gradient is then gonna pull sucrose across as a co-transporter. All right, so the last kind of active transport is called bulk transport. And I'm gonna move my face over here. All right, so here we have large molecules. These are big things that could not go across the membrane even if there was a protein involved. So these are such as like polysaccharides, large proteins, and they can't get across the plasma membrane regardless. So what happens is that the cell membrane actually changes shape to kind of wrap around and engulf things, all right? So this comes in two ways. One, exocytosis. This is bringing things out of a cell. So for example, here on this side, we have exocytosis, where we have, let's say, some waste molecules that we need to get out of the cell. And it's kind of in one of these vesicles, right? So a vesicle is just a plasma membrane sac, kind of floating around the cytoplasm. And it will fuse with this plasma membrane, and then it will release the materials. So for example, this happens a lot with secretory cells, like releasing hormones. It could be waste. It could be a number of things, right? For endocytosis, this is in, right? This is to bring molecules in. So for example, nutrients or food for the cell. So we have this big chunk of, let's pretend it's food. The cell membrane will kind of wrap around it to form a food vacuole, right? And if you remember, what would a food vacuole bind with to eat it, right? That would be a lysosome, just as a reminder, where we have low pH and enzymes, which would then help digest that food, right? So exocytosis, pretty straightforward. You're just fusing vesicles to the plasma membrane and releasing. Endocytosis can kind of come in three forms. Phagocytosis, cell eating, penocytosis, cell drinking, and then receptor mediated, which I don't even know if we're going to talk about. Let me decide. So endocytosis, again, so this is bringing things into a cell. And phagocytosis is when a cell engulfs a particle, making a food vacuole, which will then fuse with a lysosome, right? Remember, low pH, lots of enzymes, which will then be released and start digesting that food, right? Penocytosis, and the way I always remember that, Pinot is like vino, like wine, so Pinot vino. This is fluid, so sometimes cells have to engulf kind of extracellular fluid just to get what's in that fluid, all right? Making a vesicle. So this would be a vesicle full of fluid, which is not really a food vacuole, it's just like a vesicle, all right? Then we have receptor mediated, and I don't want people to get confused about this. All it is we have membrane proteins, so these little receptor is just a membrane protein. And the ligand is just a molecule it binds. So the receptor is a membrane protein that binds a molecule, which is the ligand. And upon binding, when the ligand binds the receptor, or when the molecule binds a membrane protein, that just causes the cell to, or causes the membrane to change shape and make a vesicle. And these vesicles are just different, they're fancy, they're called coated because they look at all that, they're fancy. And that's just a different way of taking things into the cell, all right? So again, for ligand receptor binding, we're not gonna go into crazy detail, but this is something that has confused people, and we're gonna talk later about ligands and receptors very often. So I just wanna say again that the receptor is just a membrane protein, well, that's it. The protein embedded in the phospholipid bilayer. The ligand is just some molecule that is going to bind specifically to that membrane protein. And that will cause the formation of a vesicle, all right? So when this ligand or this molecule binds to the membrane protein or the receptor, that just triggers endocytosis. Okay, and we will stop there. Remember, I always have practice questions at the end. Bye.